Hey guys, after we have completed chapter 2, cell biology and organization, now we are going to move on to chapter 3, movement of substances across the plasma membranes. In this chapter, we are going to talk about only plasma membranes and what is the substance that can pass through the plasma membranes and what is not. This chapter, it sounds easy, but it's actually not. This is very confusing because there are a lot of biology you guys are going to learn in the next chapters or in the coming chapters. So guys, no need to be panicked. Just go with the flow. You will master this chapter in the end. Alright, so let's get started guys. What is plasma membranes? Now, what is plasma membranes? Plasma membranes, like what you guys learned in PD3, plasma membrane is just like a line, correct or not? So this is what we call the plasma membranes. This is what we call the plasma membranes. But this plasma membrane is not actually looking like a line. If we zoom into the plasma membrane, we can see a lot of different stuff. For example, like the phospholipid as well as the protein. Now, we are going to talk about the protein and the phospholipids later. But now, we are going to look into the necessity of the movement of substances across the plasma membranes. Like, why do we need plasma membranes? And what the plasma membrane control? Plasma membranes, basically, it is the thin layer which can control the gases exchange. Oxygen can move in, carbon dioxide can go out. So this is what we call the diffusions of carbon dioxide as well as the oxygens. Now, besides that, not only gases, some nutrients, for example, like the glucose, for example, like the minerals that can move in or move out through the plasma membranes. Okay, so sometimes we have the waste products here, okay, that can be found in the cell. This waste product need to be eliminated. So they need to pass through the plasma membrane as well. So all these substances, they need to pass through the plasma membranes. All right. Now, next, the cell need to maintain the optimum pH value and ionic concentrations of the cell for activities like the enzyme or muscle and nerve activity. So basically, the plasma membranes, it form an internal environment just for the cell. It depends on what the cell needs. So let's say the pH value that is suitable for the cell is about pH 7. This plasma membrane is going to form an internal environment and maintain the pH value just for the cell functions. Next, secretes substances such as hormones and enzyme. Yup, hormone and enzyme, sometimes they can be produced from the cell as well. If the cell wants to secrete the hormones, this hormone needs to pass through the plasma membrane as well. So these are the necessity for the movement of substances across the plasma membrane. Now, what is the function of the plasma membranes? For example, like it can act like a boundary between the inside and the outside of the cell so that they can form and maintain the internal environments of the cell. Control the movement of substances in and out of the cell, as well as protect the cell. Alright, so those are the importance and the functions of the plasma membranes. Now, how the plasma membranes exactly looks like? Is it just like a line? In this chapter, we're going to find out how does the plasma membranes looks like, and we're going to zoom in to the plasma membranes. So let's say this is the cell. Okay, let's say this is the cell. We're going to zoom in to these plasma membranes and see what is inside of the plasma membranes. Okay guys, first of all, we are going to draw a little guys here. Okay, draw a little guys here, and this little guys looks like a stickman. Am I right? So this little guy's stickman, okay, this stickman is known as the phospholipids. Okay, it's known as the phospholipids. It's known as the phospholipids. Now, this little guy's phospholipid is made up of the heads as well as the tails. So the head, we call it as a hydrophilic head. Hydrophilic head. Okay, and the tail, we have another name for the tail. The tail is called the hydrophobic tail. Okay, sorry, it should be head. Okay, it should be head. So hydrophilic head. So this hydrophilic head, it has the meaning in biology. Hydro means water. Okay, philic means love or like. So this head, they are actually love or like the water. Right? They love or like the water. That's why it is attracted to the water. It is attracted to the water. But when it comes to the hydrophobic tails, hydro means water, phobic means head. So these tails, they hate the water. Why? Because this tail, they are made up of the fatty acid. Okay, it's made up of the fatty acid, two fatty acid tails. Okay, it's made up of two fatty acid tails. So these two fatty acid tails they are like oil and water. You try to imagine if you're trying to mix the oil and water together, are they going to mix up? No, they are not, because they hate each other. They hate each other, they repel each other. So water and fat, they are not going to mix together also. That's why the hydrophobic tails, they hate, okay, they repels water. They hate, they repels water. Okay, so this is the tails. Now you can see in the plasma membranes, I have drawn two layers of phospholipid. Am I right? So we have one phospholipid here, and then another phospholipid here. So we have one and two layers of phospholipids. That's why together we will call it as a phospholipid bilayer. Okay, so guys, write down. So this is the phospholipids bilayer. Phospholipid, okay, bilayer. Bi means two. Bi in biology means two. Layer means two layers. So it has two phospholipids. We call it a phospholipids bilayer. Alright, so let's move on. We're going to draw more little guys here. Okay, more little guys here. So besides the little guys, we will have one big chunk of proteins that embedded inside the plasma membranes. We call this protein as the transmembrane protein. Transmembrane protein. Sometimes we can call it as an integral protein as well. Basically, both are the same thing. Okay, so these transmembrane proteins they can pass through the two layers, the phospholipid bilayers. You can see, okay, this protein is huge, and then they pass through the plasma membranes. And you can see there's a special shape here. Okay, and this special shape of this protein is actually the receptor. This is the receptors. Now, what is the functions of the receptors? Okay, so the functions of the receptor, it is used to receive, okay, it's used to receive the hormone. Okay, it's used to receive the hormone. For example, like the insulin. Now, guys, if you have no idea what is it, you guys can try to imagine like this. So let's say this is a cell A, and then this is another cell B. Or not, it's just another cell B. Now I want to tell cell B, okay, I want him to do something for me. Let's say I am cell A, I want to let cell B to know I want him to buy the book. But I don't have a mouth. I'm a cell marker, not, I don't have the mouth. I don't have eyes. I don't have any things that I can tell the B to buy the book. So if I want the B to do something, I will send out some message. Alright, this message will travel to the B, but B, it doesn't have mouth as well. They don't have anything to like receive the message, correct? Not so they are not going to know the message. But luckily, we have a things called receptor, guys. Receptor. So this receptor, it can be found on the surface of the plasma membranes. Okay, it can be found on the surface of the plasma membranes. This hormone is going to bind with the receptors here, and then tell the cell what to do next. Okay, so for example, like this. We have a hormone here, okay, and this hormone is going to move to the receptor and bind with the receptor. All right, so this is the function of the receptors. So guys, let's move on. Now, besides the transmembrane protein, we are having something called cholesterol inside our plasma membranes as well. You draw four little guys here. In the middle, we have a 4 rings compound. Okay, we have a 4 rings compound. And this 4 rings compound is known as the cholesterol. Okay, this 4 rings compound is known as the cholesterol. Cholesterol, it is a very, very important structure that can be found in our plasma membranes. Okay, and it is also a very, very popular question. They will ask you what is the function of the cholesterol. Now, you guys can imagine that, okay, the cholesterol got two hands. Okay, these two hands can hold the two phospholipids together. So while we have the cholesterol holding two phospholipids together, what it will do, it will make the phospholipid, I mean the plasma membranes, much more stronger and stable, right? Yes, okay, that is the functions of the cholesterol. So first thing, okay, it can increase the stability of the plasma membrane. So this is the first functions. The cholesterol hold two phospholipid together. So it is able to make it more stable. By holding the phospholipid together, the cholesterol, it acts like a spring as well. Try to imagine the cholesterol is also like a spring. So by holding these two phospholipid together, it make it more flexible. Okay, it increase the flexibility, okay, of the plasma membranes. Okay, it increases the flexibility of the plasma membranes. Now, besides that, this cholesterol, it will block something to pass through the plasma membranes. Okay, so let's say there are something want to pass through the plasma membranes through the middles of the phospholipid. This cholesterol, it make it harder to pass through. The plasma membranes. That's why one of the functions of the cholesterol is to decrease the permeability. Okay, permeability. Okay, this one, remember, okay, less permeability, less permeable to what substance? The water soluble substances. Okay, they only try to prevent the water soluble substances to pass through the plasma membranes. So this is the functions of cholesterol. All right, next we have the protein, another types of proteins. I call it as a longang protein, guys. Longang proteins are because it has a longang in the middle. So this is the protein, okay, and then it has longang in the middle. This is the longang, okay. So the longang is in the middle, like this. Now, like I said just now, okay, we did study about the plasma membranes. You look at the plasma membranes, it has the hydrophilic heads as well as the hydrophobic tails. So this hydrophobic tails is in the middle of the plasma membranes. That's why it only allowed certain substances like the lipid soluble substances to pass through it. Okay, that only allowed the lipid soluble substances to pass through it. So the lipid soluble substances can simply pass through the plasma membranes. But the question is, sometimes we need another types of the substances. This another type of substances is very hard to go through the plasma membranes, but the cell needs it. How are we going to take in the substance that cannot pass through the plasma membranes? No problem. They need agent. Right? They need agent. And that agent is known as the channel protein, or I call it as a longang protein. Right? So this is the channel protein. Actually, it has another name as well. The channel protein, also known as the pore protein. Right? The pore protein. So this pore protein, they allow certain substances to go through it. Okay, for example, ions, okay, or the small polar molecule. Guys, what is ion? What is polar molecule? Polar molecule, I'm going to explain later, but I'm going to explain what is ion first. Ion is anything with charge. For example, like K+. Plus got charge, then it's known as ions. Okay, for example, like Cl minus, okay, chloride ion. So this is a negative charge, this is a ion. For example, like Na plus, it has a plus, it has a charge, then we call it as a ions. Okay, so anything with charge, anything with charge. If you guys worry about to forget this, okay, you guys write down, okay, anything with charge, okay, charge, anything with charge, anything with charges. Ions, or the small polar molecule, they are not allowed to pass through the plasma membranes. That's why they need to have the agent to help them. and that agent is called the channel protein or the pore protein. Okay, guys, so let's do it again. Besides the pore protein, we have some little guys here, the phospholipids here, the phospholipids here, phospholipids. Okay, sometimes the heads of the phospholipids is being added some molecule, okay, some molecule on top. So those molecules, they are the sugar, and they are the sugar. So the sugar in biology, okay, we call it a glycol. Okay, we have a lot of this kind of words, glycol. Glycol normally refer to the sugar. Okay, so we have the sugar on top of the phospholipids. The guys here, okay, the little guys here is called the phospholipids. It is the lipids. Now together, okay, together, the whole things come together. The whole thing come together. We don't call it as a glycol plus lipid. Instead, we are going to call these whole things as glycolipids. Glycolipids. Okay. Glycol plus lipid is called glycolipids. Then we still have some other little guys here. So we can draw it out. Okay, little guys. Phospholipid. Okay. Phospholipid. And then suddenly, okay, we have a protein, okay, in the middle. This protein is just a small one and located somewhere in the first layer of the phospholipid. Okay, so sometimes this protein will be modified by the sugar as well. Okay, so again, guys, what we call this? We call this a glycol. Right now, we call this a glycol. What we call this? This is the protein. Use your biologic. What is the name of this molecule? Together, what we call them? We call them as a glycol, okay, because we have the glycol here. And then we have a protein here. That's why I call it a glycoprotein. So this is how you recognize them. Okay, so sometimes the exam will come out like this and ask you to label it. So you guys pandai lah, okay? Okay, so glycolipids and the glycoprotein, basically they are having the same functions, okay? They are having the same functions. Both act as a receptors of hormone. Okay, so something like the receptors that I mentioned just now, it can act like a receptor of the hormone. How? Just like this. Okay, so this is a normal cell. And the cell, the surface of the cell is not smooth, you know, it's not smooth. They have some glycol, okay, protein or the glycol lipids over here. So they have a lot of these kind of things here. So it can form a receptor, and this hormone is going to bind with the glycolipids or the glycoprotein to tell the message to the cell here. So it can act like a receptor of the hormone. Secondly, this glycoprotein or the glycolipid, it can help to stabilize the membrane. So how are they going to stabilize? Now try to imagine this is the cell. It has the glycoprotein or the glycolipid on top of the plasma membrane. Now, cells in our body, they are actually immersing in the water. So this glycoprotein or the glycolipid, they can form the hydrogen bond with the water molecule. Alright, let's say this is the H2O. They actually can form the hydrogen bond with the water molecule. Okay? They can form the hydrogen bond with the water molecule, hydrogen bond with the water molecule. So this glycoprotein or the glycolipids help the cell to stabilize the plasma membranes. And lastly, it can act as an antigen. Now guys, this one is very important. This is the concept that you guys must know. So what is the meaning of antigen? Antigen basically is the identity of the cell. Like I mentioned before, this cell got no face, got no eyes, got no mouth, got no nose, correct or not? But how am I going to recognize the cell? By looking at their antigen. So we have a glycoprotein and a glycolipids over here. So they have some different shape of the glycoprotein or the glycolipids. So how the cell they recognize each other by looking at their antigens. Okay, all these things they are the antigen. All these things they are the antigens. Okay, we look at the antigens, then we know whether they are the good guys or the bad guy. Just like some of you guys are having the blood group O, correct? Not blood group O, blood group AB, blood group A, like this. Okay, why do we have blood group O, A, B, and so on? Because the red cell, it has different shape of the antigen. For example, like blood group A, blood group A. So blood group A. So it has some A antigens on top of the plasma membranes. So let's say you are the blood group B. Okay, let's say you are the blood group B. So B got a lot of B antigens, okay, on top of the red blood cell. Okay, so this is the blood group B. So how do we know, okay, what type of cell or what the cell is? We look at the antigen. COVID-19 also the same. Okay, COVID-19, they have the antigens on top of the plasma membranes, which looks like this. So this is the COVID-19. Okay, this is the COVID-19. So the bacteria, the viruses, they also got the antigens, okay, on top of the surface. So this is the antigen. Come from glycolipids and glycoprotein. All right, guys. So let's fill up the space here, little guys. Okay, fill up the little guys space here. Then we are going to draw another agent, okay, that help us to travel or transport the substances that cannot pass through the membranes. Okay, so these proteins it has no longgang. When it has no longgang and plus we have the active site in the middle, we call it as a carrier protein. Carrier protein also having almost the same functions like the channel proteins. Okay, so carrier protein, it can help to transport ions and some polar molecule. Okay, some polar molecule. Okay, so anything that cannot simply pass through the plasma membranes can pass through the carrier protein. Okay. Later, we are going to talk about the carrier protein, so no need to worry about it first. All right, so we are going to finish the whole plasma membranes with another two little guys here. Okay, so this plasma membranes is actually super thin. How thin are they? Let's talk about the phospholipid bilayer here. The thickness of the phospholipid bilayer, it is about 7.5 nm only. So if you guys got no idea what is the meaning of 7.5 nanometers, even viruses, okay, like the viruses, it also, okay, has the size like 20 nm and 20 nanometer. So you guys can imagine 7.5 nanometer, it is super thin, right? Super thin. All right, guys, I think, yep, that's it for the structures of the plasma membranes. For the first time, you guys need to draw it, okay, because it might be too confusing, okay, for you guys already. So you guys need to draw it up just in case you know how to label. So no need to draw it for the second time, just draw it once is good enough already, guys. Okay? Okay, guys, so let's revise back what we have learned just now. Okay, we have learned the structure of the plasma membranes and all the functions of the protein as well as the phospholipid. So now we are going to revise back, okay, what we have just learned. First thing, we need to know the plasma membrane is semi-permeable. Sometime in other textbook or in other reference book, we say that okay, it is a selectively permeable. Okay, selectively permeable. Same as the semi-permeable. The plasma membranes is mainly made up of the phospholipid and the protein. Phospholipid is the little guys here. Protein, it is the protein structure that can be found in the plasma membranes. So basically, just these two things. Mainly these two things. So the phospholipids, okay, it has the head and then it has tails. Okay, it has a head and it has a tails. So this is basically the structures of the plasma membranes. And then this phospholipid, they are arranged like this. It has two layers, first and the second layers. That's why we will call it as a phospholipid bilayer. Okay, we call it as a phospholipid bilayer. Now guys, one more thing that you guys need to know is very, very important. So hopefully you guys can put 1, 2, 3, 4, 5 stars, okay, in this points. It's the fluid mosaic models. So people always say that, okay, the plasma membranes, they are the fluid mosaic models. So what is the meaning of fluid mosaic models? Fluid, it means that this is the free moving of lipids, free moving of phospholipids. So what is the meaning of free moving? You look at these guys. So these little guys here, the phospholipid, okay, they are able to swap the place with their neighbor. They swap, okay, then it can become the fourth place. Okay, at first it's 1, 2, 3, 4, the third place mark, or not. But when they swap, it becomes 1, 2, 3, 4, the fourth place here. Okay, they can swap the place. That's why we call it as a fluid, because they can move randomly. For the mosaic, okay, mosaic means the protein, that protein that's embedded, okay, in the lipids bilayer. Okay, it actually referring to the arrangement of the protein. The arrangement, okay, the arrangement of protein. The arrangement of protein. So let's say this is a plasma membrane. If you look at the plasma membrane in this direction, like this, this is the plasma membranes. We look at the plasma membranes in this directions. Okay, look at this. So you will see the protein, they're actually arranged like this, okay, this like this. So let me Google for you guys to see now, okay, the mosaic patterns, mosaic patterns. Now this is how the mosaic patterns looks like. Okay, they looks like this. Okay, these are patterns, organized but not really that organized, but one box like that. Okay, like this, you can see the arrangement of the protein. Okay, that looks really like a mosaic patterns. Am I right? So one piece like that. So that's why we call it as a mosaic. Okay, mosaic. So mosaic refers to the arrangement of the protein. Okay, the arrangement of the protein.